Appreciate everyone coming here to help us think about this very challenging problem. So for those of you who are unfamiliar with Proposers Day is for you, proposers, right? So it is a time for us to describe the problem in as many ways as we can so that you're clear on exactly what we want. It is a day where we help you understand exactly how to apply. I mean, this is critical. We know that applying to the government can be, technically there's a lot of registration, there's a lot of getting things aligned. I really encourage you in a little bit to listen to Adria, because if you are 10 minutes too late in your submission of a proposal, an abstract, 10 minutes too late is too late. Right, so please pay close attention to Adria. It's also an opportunity for you to find other partners, right? And so that's the whole point behind lightning talks and posters and the coffee breaks, is that you can talk to each other, because we're going to ask you to pull all these pieces together into a coherent team. So it is a great place to partner up, right? So we will also be answering questions throughout the day. We will be taking questions through the IGoR website, so you can send your questions there. Our IGoR email address, igor@arpa-h.gov. So you can send them there. I think we will also have some paper slips if you want to write down your questions and then hand them to Renee at the front desk. And then lastly, just moving over to the program itself. I mean, IGoR is a very ambitious program. I think to really understand the program, you have to know something about program managers. So the thing about ARPA-H is they go out and they find program managers. As a program manager, I like to say they look for bold people. Probably more accurate description, they look for arrogant people. Arrogant people who think that you can ask questions like, hey, can we just redo all of science? Can we just, reorganize the entire scientific endeavor to make it more efficient? Let's just pull it apart, pull all those pieces back together to make it more effective, right? And so we're going to pull the pieces apart. Actually, you guys, you signed up for it. Your name, you already registered, so you're in for the IGoR effort. How we're going to rework science. Wrap all these capabilities around the scientist, the researcher, to go faster and ultimately to develop new therapies even faster for the patients who need them. Okay, so with that, we have the distinct honor to asking Dr. Alicia Jackson, the director of ARPA-H, to come say a few words and to kick us off. Alicia.
Thank you all for being here, and for everyone online who's here. I really have to thank Paul. Paul's the real star here. My job is incredibly easy, which is I get to work with incredibly bold program managers and support them. That it's such a pleasure. I can't even begin to tell you. And exactly what Paul said. Who could be so arrogant to believe that you could go and pull all apart of science and rebuild it again, but in a much more rigorous, reproducible, and faster way? And I would say this program is really ARPA's ARPA. Our entire mission is to be able to drive breakthroughs in years and not decades, to really act as a time machine. And I think all of us who've ever worked in a lab as a scientist and engineer have felt that pain of running an experiment, getting a weird result, trying to reproduce something from a laboratory, maybe just down the street, it not working, literally calling up the graduate students in the lab to ask them what they did that maybe they didn't publish in the paper where they ordered their reagents. And that doesn't scale. And it's very frustrating, I think, for many of us to see an incredible result published in a top-tier journal, and yet it won't reach a patient for 15 to 20 years. I think it's even more frustrating when you read in the literature that there had been an error, a mistake. One recently came out in the longevity field. People had mistakenly ordered some of the wrong antibodies because its name was really similar to another one. And so suddenly a lot of people had been running experiments using the wrong antibodies. And that just slows down all of scientific research. And so we're at a really unique time right now, I think, in the scientific enterprise. Cloud labs have been around for 15 plus years, and many of you know them. Number two, we have an incredible wealth of knowledge now available across the world. Number three, we have this thing called AI, which enables us to really go read across all of that knowledge. And the fourth thing we have is we are starting to see this tie between AI and automated experiments. And we see it happening in silos. We see the benefits and limitations of all the different components of this. But I think what Paul really sees, and I think many of you in this room see, is now the building blocks and the components to really stitch these together in a really radical way so that we can expand not only scientific knowledge across every lab that wants it, but be able to work collaboratively to be able to work on a system where you can reproduce experiments. Importantly, you can understand, even if it's not your knowledge domain, what's an experiment you might need to run next, a gap in the knowledge, and then be able to put it out to a marketplace and have it run in a high quality way and be able to run it over and over in multiple places and have that data actually reproduced. And that's a pretty bold vision for how science could run. And it's also incredibly democratizing. We all know what universities are like, are very, where you have kind of the professors who built up these giant research teams across many different domains. They have tons of equipment, and they can move at a pace that, let's say, a young professor or professor at another university simply can't. And so now we can start to come to the point where really the best ideas can win. And that's a really exciting thing. And it's an exciting thing for us as an agency because our entire job is to go find the people with the best ideas and to empower them. And at the same time, it's also incredibly empowering because the number of innovators and brilliant people we have across the United States is unlimited. It is truly huge. And being able to give people the resources and the tools to be able to take those ideas and act upon them, that young graduate student to be able to give them those tools and act upon them is incredibly important, especially if we're going to tackle so many of the challenges that we're facing today. For those of you who haven't worked with ARPA before, buckle up. You're going to have the greatest time of your entire life. I like to say that being a PM is actually the greatest job in the world, so maybe you'll become one of those. But really, nothing happens in an ARPA without people working together. We have no internal labs, and that's kind of a superpower for us because it means the only way we're successful is if you're successful. We are all on team together. I know that there's 3 interdisciplinary teams that are going to form around IGoR. And not only will that require incredible teamwork within those teams, but also across those teams, being able to share experiments and work together. This really is a team sport altogether. We want all 3 of those teams to be incredibly successful, and anyone who's around and within those teams also individually to be successful. We can't do it without you. You are actually the missing piece of all of this. And it's incredibly important to the success of the program that not only you come to the table with your best ideas, but you help recruit the best people you know to come to the table with their best ideas. And this is the first step to meeting one another and getting to know one another. But I know there's tons of people online as well. And so this is really the moment. We do try to move very, very quickly because we don't like to wait around. We don't want to wait a year to actually act. And so I'd highly encourage folks to find others who are deeply excited about this space and to be able to work together. I can't say enough about Paul and Yoram for stepping up to lead this. It's a massive effort. It's very exciting. And if it's successful, it really does transform how we do science and drive development in the United States. And across every disease areas, we do a lot of programs that maybe focus on one particular problem or another, but this is really the enabling infrastructure. And the closest analogy I can come to is a little known agency called DARPA, who built something called the ARPA-Net, which it wasn't like people didn't have computers and could do their own experiments on computers. It was really being able to tie those together and have protocols to be able to share that information. And we didn't know what it would lead to. But it led to exactly where we are today and enabling potentially this revolution in science. That's a very bold claim that this could be that, but I think many of us understand that if we can unlock what we think we can here, this ability to move science forward incredibly quickly, to understand what experiments we can do and to be able to reproduce that data, I think it feels very intuitive that we should be able to unlock a similar revolution. We can predict some of those benefits, but there'll be many, many more on top of that. Finally, I'll just say, we're always recruiting at ARPA. I spent 5 years of my life at DARPA. They were an incredible 5 years, first as a program manager and then as a deputy office director. And I always said, if I were to ever come back to government, it'd only be for an ARPA. And I went off and built companies, and that was fun. But there is nothing that lights you up and fills your soul and know that you are working on behalf of the American people like working at an ARPA. You are truly unleashed here. In a company, in a lab, you are always constrained by resources, constrained by having to meet your profit or revenue targets, constrained by having to publish that paper. Here you are unconstrained. You can go work with the best people in the world. You can bring them together to work on ambitious efforts like this. And so if in the back of the mind you're thinking, geez, this sounds really exciting, please come talk to us. We are looking for incredibly bold people, a little bit arrogant, but importantly, we don't ask our program managers to have the best ideas. We ask them to find the best ideas. And many of you have incredible ideas in your head right now or know people who do and see a future that we could build together. It is an incredible privilege, and it will, I promise you, you will never have more fun in your life than being at an ARPA. So with that, I'm very thankful for all of you being here. We really do need you. We need your best ideas. We need all of your efforts. We need you to bring the right people to the table, whether they be companies, academics, nonprofits, whoever they might be, and really show us how we can build the future. Thank you so much.
I love the way Alicia talks about science. I kind of want to apply for my program now. There might be a conflict of interest lurking in there somewhere. Okay, so I think now we're going to step through overview of the effort. So one of the things about being an ARPA-H program manager is that we're term limited. We come in, we get something done, and we move along. So I'm going to be moving along over the summer, and so actually on. For me, while I was at DARPA, is the inspiration. So we're going to tag team a little bit of the presentation. I was going to introduce it, and then some of the TAs where he has amazing expertise. I'm going to have him step through. Okay, so maybe, and I'm in control up here. Yeah, I think you've seen your agenda. We handed out copies. And just as a reminder, we're stepping through there. We will be talking about acquisitions later. If you have questions, please continue to send them in. We will handle the acquisitions questions live right after Adria, and then we'll have the technical questions at the end of the day after the lightning talks. All right, so let's start talking about IGoR. So what if we could make biomedical research ideas earlier? This is, per what Alicia was saying, an experiment might require another lab's instrumentation. One of the questions is like, hey, I'm studying some rare disease. I run, facts, I run a lot of omics, and then the AI, I'm analyzing the problem. The AI says what you really need is cryo-electron microscopy of, these two molecules binding. I don't do cryo-EM. Nobody down the hallway does cryo-EM. How do I find somebody who can do that in the right way and provide the information back to me, right? How do I prevent those dead ends? And then lastly, right, published results of poorly documented protocols. I've written about IV&V and trying to ensure well-developed protocols for years now, right? And so it holds things back. And I don't think this is nefarious, right? But oftentimes someone's ordering the wrong reagent. That cell line has drifted in phenotype, something happens, right? And then suddenly the experiment isn't reproducible, right? And so the consequence, you pull all these together, research is slow, it's often irreproducible, it's difficult to integrate into working an effort, right? And so decades can go by. This has a real impact. And these, there are more numbers I can throw at you, but these are all terrifying, right? And so just looking at publicly funded research in the US, 70% of researchers have failed to reproduce. to try to do it again, and 70% of people have said, like, yeah, I never got the yield, the cells never did that, I never got that signal when I try to reproduce this important research. 85%, and this is a number from 2015, 85% of biomedical research efforts are likely wasted due to poor research focus, flawed study designs, and publication bias. And that ends up, again, this is 2015 dollars, so it's even higher, estimated $28 billion spent on preclinical research that's irreproducible. And all of this, help leads. And so our goal, your goal, the goal we're tasking you with, is trying to speed up research so that those patients can get the therapy that they need. So the way we're doing this is we're wrapping a set of capabilities around researchers, right? I know many people are, very interested in self-driving labs. I'm not. I like research. I love research. I think people should be doing research. And so what we're doing is bringing tools surrounding the researcher so that she can be more efficient at what she is doing, make progress more quickly. 4 main components, they all fit together in a virtuous cycle. So one aspect, we're trying to build out models of disease. These digital twins, they're going to be mechanistic, and that's going to enable the researcher to reveal obscure connections. Second part of that is, okay, we have a model. How do I identify what is the next best experiment to be done? So it's a collaborative work between the AI and the researcher to develop new science, explore models, develop new hypotheses. Then we get to what I think is going to be one of the most challenging aspects. Okay, I have an experiment that needs to be done to inform this model that I'm building out. How do I write a procedure that is exact, that can be done either in my lab or in some other lab or in a marketplace of research? So we're going to come up with these interoperable procedures that convey the essence of those experiments without the researcher having to write up a giant protocol each and every time. Just having those protocols that can be sent out doesn't help if you have no place to send them. So the last bit is we're going to stand up a marketplace that can respond, that can take in these protocols, perform the experiment, and return data back to the model. And the idea is that this all happens very quickly, right? You can write up, you can look at your latest results, you can identify what the next most important thing is, and then you can send the IGoR out to fetch more data the next day, the next week, and you can get it back very quickly, hours to days instead of months to years as it is now. Ultimately, this leads to faster insights, leads to much broader access to our experimentation marketplace. People are not going to get shut out because they're not at a giant, incredibly well equipped university or company, and ultimately novel therapies with lower development risk. So this is all like, how far will science go with IGoR? Pre-ChatGPT, models were sort of purpose-built. Where we are today, people are having AI research agents as research assistants. Where IGoR is going to take us is these AI agents as sort of multimodal integrators. Scientists and the agents are going to collaborate to explore this rich landscape. One of the reasons we like the mechanistic is ultimately we think all these pieces with time are going to build up. And so ultimately maybe you have, complete digital twins of people, of drugs. And so kind of human judgment applied to really unbounded knowledge. So it's a system and not just sort of a one-off. A little bit of guidance. So one of the things that, we really want to impress upon people is that we are funding this to the point that you should not be looking at toy problems. For reals, you're going to go out, you're going to grab enough data to make progress towards solving a disease. So we're funding it to that limit. So it's not just don't go pick an easy problem. We're going to go after an ambitious problem and show that this process works, right? So you are going to propose your own complex human disease to study. In the proposal, in the abstract, briefly describe the state of the understanding. It should have a broad distribution or be, highly impactful. Ultimately, it should be addressable at the cellular or multicellular level. I'll jump into that in just a second. And it needs to be tractable via mechanistic modeling. So under the cellular multicellular level, right? So IGoR is going to advance AI-enhanced research, but we are not going to have the AI direct human or animal trials. right? So within scope are diseases that can be addressed at the cell or multicellular level, organoids, lower invertebrates, right? What is out of scope is we're not going to have AI direct human clinical trials or studies on vertebrate animals. Note that if you already have existing human samples, that can certainly inform the model, just as, reading the paper, that is fine. We're just not going to have AI directed research. Similarly, vertebrate models can be used for confirmation, right? If the researcher, with their own judgment, decides that this is what is needed to confirm results, that is fine, but the AI is not directing that. Models need to be multifactorial. They need to have dynamics, and they need to be multi-scale, right? Go from intracellular, subcellular to multicellular scales and multiple time scales. By phase 3, this is a process. We want this to be extendable to a related second disease area, right? So I say all these things because it impacts like what disease you're going to go after. I know everyone has their favorite disease, but just make sure that what you go after is addressable in this manner. Okay, Yoram, do you want to talk about TA1 and 2?
Thanks, Paul. Before launching into this, I think it's important to give just a little flavor of my own personal background. Some of you may know me more for the work on computational modeling, but when I was a postdoc at NIH, I had the privilege in the early 90s of having access to an automated immunostaining robot that gave me, as a single individual postdoc, the ability to do a large amount of staining. That was great up until I had to deal with coming up with what the original experiment should be, and also how to interpret and deal with the results and how to repeat the outcome. And while that didn't directly shape where I was going, another piece that did shape where I was going was realizing just how complex biology actually was, how dynamic it was, how nonlinear it was. And that very much informed my segue into organically sort of into computational modeling a few years later. And that is really a big piece of what's in TA1. So on the one hand, as Paul just said, the rubber will meet the road in cellular, multicellular, or organoid type models in IGoR. But we want you to be thinking in the concept of a digital twin of disease. So it would probably be something to consider in the back of your mind that, as a very minimum, some of the models that you're going to be developing in TA1 need to at least connect to other higher order models in some way, either as one big unit or in some other fashion, so that you can convince us that the work we will be funding will, while it will play out in a system that is decomposable down to cells or tissues or organoids, there is direct or fairly direct pathway to applicability to an actual disease. So you will need to develop model architectures, so establish languages and schemas. You will need to be populating these with known biological data, and importantly, identifying the knowledge gaps, because the point of this is to lead into the next technical area, TA2, where you're going to be trying to do that next experiment, as Paul said. So I'm going to reiterate this just to make it very, very abundantly clear. We're not looking for LLMs, correlation-based models, et cetera. There is certainly room to consider hybrid type architectures, but the models have to be mechanistic in nature, have to be dynamic in nature, have to be simulatable, right? You have to be able to play them forward in time, make predictions. They have to be modular, multi-scale. They have to be verifiable. And as I said, they have to at least have that ineffable aspect of convincing us that you have an idea of how this will ripple forward in scales up to the human disease level. TA2, we're calling it the new science engine, but you could also think of it as a digital twin of the scientific thought process. It's more than just about a decomposable simple hypothesis. It's about how is it that an understanding computationally in TA1 of a disease and thinking of it in a cell context, how will that lead to that next experiment? And of course, a hypothesis should be what drives that next experiment, but it's that connectivity of the thought processes that leads to that hypothesis that I think is where the really interesting new insights will come. So the idea is that this needs to explain model gaps. It needs to be able to tell us that, yeah, the model is good enough to make this, to get up to a point, but in order to be able to connect, let's say, to that next level that gets us to a real understanding of a disease, there's a gap. Okay, that gap is the new piece that needs to be tested, right? That is where the hypothesis will show up. But understanding the gap is more than just the hypothesis. As Paul said, we're not looking to replace human scientists. We're looking for this to be a sort of human AI teaming, co-generating hypotheses. And so there needs to be an understanding of how to iterate with the researcher on these hypotheses to be able to fill in those gaps, those blanks. And then ultimately, it needs to, all of that has to sort of be made practical, be made real by creating experimental plans. And that's the part that will then feed into technical area 3. And I'm going to move that back to Paul.
Thank you, Yoram. Okay, so once you've hit TA2, you should know what that knowledge gap is, right? And you have an idea of what needs to be done. And so the next part is, how do I go out and get that experiment performed and get that data back to me, especially if it's something that I can't do within my lab. I don't have that cell line. I don't have that instrumentation, right? I actually think that TA3 is going to be one of the most challenging aspects, where the researchers should be focusing most of their time. From then, you're going to generate, from there, the system will be generating procedures, multiple ways of going about this, using existing libraries, procedures, mapping from the knowledge gaps, trying to understand what is the best experiment to be performed and how that experiment should be performed. It can optimize. So one of the things, aspects that we're going to get to by the end is you should know, okay, I want an experiment to be done. You're going to need to know which labs can do it. How much is it going to cost? How long it's going to take? And it's not within the program, but you can start thinking about, like, okay, with how much noise, right? Because a lot of times people can spend a lot of money on beautiful spectra or very detailed results when the AI just needs to know if there's a blob somewhere, right? And so one can actually think about that. It's experiments. This is going to be a road that we walk down. We expect everyone to be, who is funded, to work together. We're going to have working groups on this. We're going to be working with NIST to help us identify calibration standards, calibration approaches to do this approaching. We're going to have everyone who's a part of this is going to have be a part of the groups taking feedback, right? Different requests. They're going to be bake-offs, and we'll work through this. So put forward your best idea, but know that this is going to be a collaboration among all the teams to work through this very tricky problem, because ultimately, if we want a marketplace, we're going to have to come up with some common standards, right? So this is tough. Bring us your best ideas. We think that this is a good starting point. We're open to other processes, but that's TA3. So TA3, we have this system whereby we can request experiments. Doesn't help if we don't have a marketplace that can fulfill it, right? So it's one thing if you have a friend down the hall, a collaborator at a different university, but here we want to open it up. So the way we're going to get to this experimental marketplace is stepwise. So what's going to happen is that in phase 1, every team that comes forward, and look around to the people here, every team that comes forward is going to have two aspects. One, they're going to have the requesting researcher, and for every one of those requesting researchers, they're going to need to have at least 2 labs that they're working with that can perform experiments and send results back, right? So the expectation is on the order of about 3 teams, each bringing at least 2 labs with them. You can bring more. You can bring as many as you like, but at least 2. So that's the in phase 1. Show that you can request those experiments. Show that the data is getting back to the researcher correctly. Show that those 2 labs that you're working with, right? If I ask the same experiment to be done with both of them, then I'm getting the same result, right? So that's phase 1. Phase 2. As we're looking at reproducibility and interoperability, can I start to request experiments from other labs? So, if I'm doing some, FACS analysis or, some spectroscopy in order from my lab and this other team's lab, am I getting the same results, right? If not, those working groups are going to have to work out it, get on the same page. So that's phase 2. And then phase 3, we're going to have this proto-marketplace, prototypical marketplace, early stage. Anyone can order experiments from anyone else. At that point, The hope, the intent, the plan is to have these standards, these protocols well-defined enough so that if other people want to come in who are never even part of the program and say, yes, we can hit those marks too, there may be a broader marketplace in phase 3. So that's the plan, stepwise, to build up this marketplace so that at the end of this, anyone can order experiments from this marketplace. So, some notes like what IGoR is and isn't. Proposers Day is a great time for us to try to clarify, right? So what it is, it's mechanistic, it's multi-scale, digital twins of disease. The system's going to act like an AI orchestration layer that augments the scientist, right? We're keeping the scientist in the middle of it. It's open. We're going to generate these open layered protocol stacks governed by community standards. Right? This is going to be a collaborative effort, and ultimately, we're going to lead to a distributed, validated experimental marketplace. So what is not? It is not a frontier LLM attached to an automated lab, right? You'll be discouraged from continuing on for a full proposal if that is forwarded. It is not an AI that replaces the researcher or acts irreversibly on its own. Right? It's collaborative with the researcher. It is not closed. It's not proprietary. It's not a single lab demonstration. As you can see from the description of TA4, stepwise building up of this marketplace, and it is not narrow optimization against a single objective. Do not come to us with a toy problem. Come to a tractable problem that you think that this approach can solve. All right, so. All the key outcomes will be enabled, but it's really at the interfaces. So we break these things up in TA1, TA2, TA3. We know that how these different aspects are glued together are just as important, right? So how TA1 connects with TA2, this design of experiments, the enabling of the design of experiments beyond institutional capabilities, that's between TA2 and 3. Let me start all the way on the left. Being able to reveal these obscure connections and ask high impact questions, connecting TA1, TA2, between TA2 and TA3, being able to design experiments beyond your institutional capabilities and talk about that in a reasonable way. This ability of, I have this procedure, but how do I get it to the marketplace? How do I order reproducible experiments with standard protocols? And then even returning that data, right? We have to ensure that all the appropriate metadata, all the appropriate details are there so that when this is sent back to advance that mechanistic model, you have all the data needed to do that correctly. All right. Next up, we have John Elliott from NIST. We've been talking to them deeply about a lot of their thinking, a lot of the work on how to create standards for experimentation in the biomedical research enterprise. So over to you, John. There's your clicker.
Thanks. Thanks, everybody. My name is John Elliott. I'm from the National Institutes of Standards and Technology, which is a part of the Department of Commerce, another federal agency. A very unique one, though, in that it has a unique mission and that it focuses on what is, I'm going to call measurement infrastructure, which is an interesting thing. And the title of the talk is Measurement Infrastructure for Distributable Assays. A whole bunch of interesting words there, but I am going to try to highlight what does that actually mean technically, and what do you do to develop distributable assays that can be ran at different locations. You can get the same answer from different locations, and you have the ability to have confidence in the quality that the assay answer came out correctly. So I was going to give you a little bit of insight into those challenges and what is required. So NIST, which is the National Institutes of Standards and Technology, we, again, are the National Measurement Laboratory. We focus on this measurement infrastructure, which includes things like reference materials, new measurement technologies, protocols, reference data, a variety of things that help improve your ability to make measurements. I look at the challenge, kind of the grand challenge is how do we get, we both have the same sample, if we share a sample, how do we both get the same measurement in each of our laboratories? What are the details of that? Just to show you how interesting measurement science is as a activity, is the lower part of the slide where it says components of a measurement. Measurement science really breaks out a bioassay into one thing. It's a measurement and it's got three components. One is value. That means it has a number. It's on a scale. So you need to figure out a scale that allows you to take two measurements and put it on the same scale. It has an uncertainty associated with it, which allows you to tell whether the number you're measuring now is statistically different than the number you measured last time. That's about replicates and those ideas. And then the third thing, a really critical thing, is evidence that your measurement system is working. Any measurement system can give you a number, that can give you uncertainty. The question is that the right number that's coming out of it? So those three things are what is critical to think about a measurement that's going to be useful for a distributed assay that can be performed at different laboratories. So next slide, please. So why am I here talking about this? Of course, as we saw in the last presentation, there are two kind of activity areas that are a critical component of the IGoR program, and that is shown here in blue, the standardized protocol architecture that allows scientists to perform these experiments reproducibly. And then the second thing, this marketplace of qualified laboratories that can execute these protocols. So if you look at those there and through a measurement lens, I see two things. We need measurement infrastructure for high quality measurement systems. That really makes it for a good assay that's reproducible. And a transferable measurement system, which means that I can transfer somehow the idea to another laboratory. They can run it, and I have confidence the number that I get back is actually the right result. So go to the next slide, please. Let's talk about high quality assay. What is a high quality assay? At kind of the way we think about it at NIST is interesting because it breaks apart a really important component of biological assays apart, and that is separating technical validation concepts or technical analytical validation from physiological relevance. The measurement is about the technical validation. The meaning of the measurement is not of interest. Well, it's of interest, but it's not necessarily integrated into the ideas of how to build a technically valid assay procedure, which has, again, scale, uncertainty, and has evidence components in it. When you have both of those pieces, you have a high quality assay and you have an adverse or some kind of physiological relevance pathway, which again is kind of part one of this project. It allows you to build physiological assays that have high quality, so you have confidence. Here are some of the tools that you use, and this is maybe new to a lot of people. These are engineering tools that have been adapted over to biology ideas. One is a cause and effect diagram. One of the most important things to do when you have a measurement or an assay that you're going to then think about having other places to be able to run it is sources of variability. What causes the wrong answer to come out of your assay? And that is what this tries to map out. There are different ways to think about those sources of variability. Importantly, sources of variability are shareable, and that you may be running some similar assays, different endpoints, but some similar assays. So information you learn about one part of your assay can may be useful in the next part. So cause and effect allows a way to coordinate sources of variability. What is nice about the plate design is you have an entity that is represents the assay. The plate design can encode control experiments, can control the experiment that you're actually running on it. All of that information is encoded into this plate map, and those plate maps are important ideas to make sure that you can have comparability and a variety of different control measurements built on with your assay. Again, provides confidence ideas. Next slide, please. The other two things that come up often when you're really looking at measurement infrastructure is the idea of control charting. That is, whatever your control measurements are in your measurement system, you are tracking that over time, and you know when your assay is starting to fall apart because you can see it is coming out of spec. For assays is really critical to move to understanding how your assay performs and when it's not performing correctly. And then finally, that leads to is specifications, and these are really important. Once you have specifications, you then, which could be based on several things, control charting, you then have the ability to perform an assay, look at this, your control measurements, look at the specifications, make sure the controls fall within the specifications, and then you have confidence that the measurement system worked right. So the idea of specifications is really important. Next slide. I didn't talk about automation yet, but as you can imagine, this really heavily ties into the distributed manufacturing ideas, cloud laboratories, biofoundries, the idea of having a high quality assay that can be performed at different sites. So I am going to talk a little bit about that. There's some ideas here, but certainly a really interesting part of this project is being able to have a marketplace of people that can do assays. So I am going to talk about this automation friendly. All of these ideas I've talked about are automation friendly. One of the things that I've found useful is to think about an assay plate design and the specifications for that plate as a product from a manufacturing process. So you are manufacturing an assay plate. That really allows you to look at a variety of tools for process engineering. One of the big ideas is there a product-based minimum performance specification for an assay? If there is, what is great about that is it's based on the plate design, not on how it's made. So anybody with any robot that can meet the specs can make the plate. They can perform the assay, ideally. And so in theory, the idea of performance specifications for a plate is what begins to enable the ability to perform the same assay at different sites. All of those ideas, 100% automation friendly. These are very fundamental manufacturing ideas of using performance specifications to get the products you want, and LLM script friendly in that they are product based and it doesn't matter what script you had to run it as long as it generated the right plate. So these ideas are really important for automation. Next slide. And what you might be able to see in the future, and this may be a product of this may be what your measurement looks like coming out of the IGoR, is you have developed an assay on your automation system, works really well. You have done enough work where you have specifications. You build a standard that here's my assay plate, here are the specifications. You give that to someone else. If they can perform and show you that they get that assay plate with those specifications, they can run your assay. So those ideas, it's not fully developed yet, but this is where we are in trying to understand how to facilitate this. Next slide. One last slide, I put this in just because the explosion is heavy. Biomics data, whether it's combined omics data sets, they are coming like wildfire, and they are very complex data sets. So all of these ideas I talked about will be important in these omics assays or any kind of multiplex assays. You've got to be able to have specifications to ensure someone can do it. So how do you do that? It's a research project right now. There are some ideas. Again, we frame everything as a sensor array. There's a variety of tools for sensor arrays that are interesting. So we can begin to adopt some other technologies to enable us to build shareable biomics assays. And again, a really important idea of this is that you do that separate from the physiological analysis of the biomics assays, and that the meaning of them is the second part of the assay. The first part is making sure the measurement quality is good, and that the measurement quality was appropriate from another site that did it. Thank you. Next slide. So summary, I've highlighted in vitro ideas about measurement quality, transferable assays. Like I said, technical validation, everything I've talked about is not biological validation. It's not about the disease. It's not about what the assay tells you. It's about what the measurement is and knowing about your measurement and your assay. There's a variety of tools available that will be important. And I just going back to looking at what the IGoR call out is, high quality assays require calibration, replicates, controls. Measurements can be used to build these minimum performance specifications. That may be a strategy to transmit your assay needs to another location. Assay requirements, as I said, these are specifications are how assays are communicated through these bio networks. So it will be important to establish those ideas. Ultimately, you can imagine these lead to standards development. Maybe your assay and the specs are a standard for running that, and that's has a tremendous value for someone to use. So that is my summary. I can take questions or later. If you find me, please ask me questions. Next slide, please. Further contact, my email is there. We have a great measurement assurance website with a lot of ideas and things to look at. Yeah, let me know if you have any questions. Thank you.
Thanks, John. It's now my distinct pleasure to introduce Adria Robinson, who's going to talk about the acquisition process with ARPA-H for IGoR.
Hi, good morning. I'm Adria Robinson. I'm the agreements officer supporting Paul and Yoram on the IGoR program. Let's see, am I running or are you guys driving the slides? Okay, next slide. Right, the awards under IGoR will be other transactions. We'll talk a bit more in a moment about what that means. We are not setting funding limits or funding ranges for the award, so please don't ask us. What we are asking you all to do is to build your solutions, price them appropriately, and then we will evaluate accordingly. Solution summaries, right? So this is a two-step submission process, solution summaries being that first step. That submission is mandatory. You cannot submit a proposal if you do not first submit a solution summary. When you submit your solution summary, it will be reviewed and you will receive a letter from ARPA-H stating either we encourage you to submit a proposal or we discourage you from submitting a proposal. You can submit a proposal regardless of which way that goes. However, I highly encourage you, if you receive a discourage, to consider whether or not it makes sense to submit a proposal. If we discourage you from submitting a proposal, what we are telling you is that it is not likely to be successful. So seriously consider whether or not you want to spend the money and the resources to put together a proposal in that instance. The last note on this slide is something I am going to say multiple times while I'm standing here. Read the ISO and then read it again. And then you should probably read it a third time. All of the information you need is there. If you feel something is missing, feel free to ask us, but don't reach out the morning that submissions are due and tell me that you need help because you chose not to read the ISO. Read the ISO. Don't assume that it is the same as any other ARPA-H ISO. Don't assume that it is the same as an RFP or a NOFO or any other solicitation that you might have seen from another government agency. This ISO is specific to ARPA-H and it is specific to this program. And I promise if you read the ISO, you will have what you need to be successful. Next slide, please. I want to show, this is a screenshot from sam.gov. This is where the ISO is posted, and this is where any amendments to the ISO will be posted. If you look in the upper right, where the arrow is pointing, if you log into SAM and you navigate to IGoR, click follow, so you can receive notifications if we post amendments. I don't want you to miss an amendment, right? A document being updated or additional information. So this is a way for you to get those notifications directly from SAM. Next slide. I also want to show how SAM, how it looks when we amend. So if you look at the drop down there, you will see that there is a May 5th date. That was the original posting. And then there is current record. So that was our first amendment. So whenever you navigate to IGoR in SAM, if you want to see what the latest is, you want to click on current record, but you can always click that drop down and look at any of our previous postings and download older documents or see what our notes were. But SAM does not, it doesn't very clearly show you, hey, this is an amendment. So this is how you will find the latest version of the ISO. Next slide. All right, other transactions. Other transactions are agreements. They are legally contracts, right, between ARPA-H and whichever of you receive these awards. They are not contracts in the traditional government contract sense, right? So they are not bound by the Federal Acquisition Regulation. Other transactions are not grants. ARPA-H does not award grants, so they are not subject to the financial assistance regulations either. The other transactions allow us to negotiate more freely with you all, right? It allows us to look at your proposal and decide, is this going to fit the program without some of those regulatory constraints that the other awards have? We can negotiate with you more like a commercial entity would negotiate than traditional government. So one of the things that you will see in the ISO that's posted is that there's an appendix, and it is a draft of the terms and conditions, a draft of the other transaction. And one of the things we're asking you to do as part of your proposal is read through that and provide us with questions, comments, or red lines, right? You can propose edits to those terms and conditions that we will then discuss and negotiate with you if you make it to that stage. So that's the wonderful thing about other transactions, is it allows us a lot of flexibility, and it will allow you all to work in ways that you are used to working without having to conform to really strict government regulations. Next slide. Everyone is eligible to submit as long as you are not a U.S. government entity. So that includes agencies, that includes FFRDCs, that includes those entities that identify as quasi-government agencies. Okay, you are still a government entity, so you are not eligible to propose. If you are considering having one of these entities on your team, reconsider. They cannot be a part of a proposing team. SAM.gov, that is where we post the ISO. That is also where you are required to be registered in order to do business with the government. If you are registered in SAM, you need to check that your registration is current. SAM registrations require annual renewals or updates if there are material changes. This is incredibly important. The SAM registration process can be very smooth for some people and very difficult for some people, depending on the type of entity and the documents that you submit. There are people who get through this process in a week. There are people who take months. You need to start now. If you call me the day before proposals are due and tell me that you just started your registration process and it's not going through and you don't know what to do, I'm going to feel for you. I will. And then I will reject your proposal. The SAM registration requirement does not apply to your solution summaries. Okay, it applies to when you submit the proposal. So you do have a little bit of time, but let's pretend that you don't. I cannot, under any circumstances, award to someone who is not active in SAM. And there is no waiver or exception to that. I am bound by that rule. So this is really important. Next slide. All right, so the first step, as I said, is submitting a solution summary. It is mandatory. We have provided you with a template at Appendix B. These are reviewed and not evaluated, which means we are not making selection decisions. We are going to give you that encourage, discourage feedback. The next step is submitting your proposal, and you can only submit a proposal, again, if you've submitted a solution summary. We've provided guidance at Appendix C. There are templates at appendices C1 through C4. And the evaluation criteria, which I will flash up on the screen in a moment, is how we are going to evaluate and make selection decisions. Your submissions are reviewed independently, evaluated independently, so we don't compare you all against each other, right? The only comparison is to our evaluation criteria. Once the proposal evaluation step is done, we will notify you as quickly as we can about our selection decisions, and those of you who are selected will receive instructions at that point on how we're going to move forward with negotiation and award. Next slide. All right, so these are the evaluation criteria. These are in the ISO in more detail. They are listed in descending order of importance. We are going to provide these slides, just so you guys know. It's fine to take pictures, but I just want to let you know you will get copies of the slides. There's a lot of photographers in the room for those of you in the front row. So this is how we will evaluate your proposals, right? We stay within the four walls of these evaluation criteria, but this will help you understand how all of the instructions we give you then feed into what we're going to do with your submissions. Right, next slide. So here we're just kind of laying out the different documents. At the solution summary stage, you'll have a cover page that is already a part of Appendix B. We do ask for a rough order of magnitude budget. We are not looking for a lot of detail in the budget. We really want just to have an idea of where you think your solution falls in terms of how much it's going to cost. You are not bound by that number, right? You might have a different number in your proposal submission, but if it is wildly different, then you need to make sure that in your proposal, everything is very clearly explained. In the proposal, right, if you've submitted to an ARPA-H program before, you know that sometimes you have to submit very large, very detailed, very complex pricing spreadsheets. I don't want those, okay? So we have provided you with a template. You will see that there is a lot less detail than we typically ask for. Please don't email me and ask me if I want more detail, because no, right? Just give us the information we need. We are trying to make this process easier on you by only asking for what we think we actually need. If we find that somebody submits something and we select it and we do need some more information, we will discuss that with you, but we only need what's asked for. I want to call out Appendix D, the Administrative National Policy Requirements document. This is, all of the documents are important, but this one is very, very important because this is where we cover things like biosecurity, research security, and so forth. There are going to be attachments to this document. So please read this document thoroughly, right? You will need to submit biosketches for key personnel. You will need, there are signatures required and so forth. So please read through this very carefully, because we cannot have anyone working on an ARPA-H program who has not gone through our research security process. And if we don't get the information we need, we won't be able to complete that process. Next slide, please. Teaming. Today is a perfect opportunity to find team members if you are looking for some. We do have a teaming page. You sign up, you can search, right? You can search on the teaming page, and Yoram is confident that it's a yes. Okay, so you can search the teaming page to find partners and start building out your team. It is likely you all are going to need to form teams because you need to be able to respond to the totality of the program. The way the awards work, however, is the prime or the lead proposer. That's who signs the award with ARPA-H. And then the other members of the team will sign contracts with that entity as essentially subs. That might be my last slide. Next slide, please. Final guidance. All right. The ISO is binding. So if we say something today that conflicts with the ISO rules, right? If you get an answer to a question that conflicts with the ISO rules. We do try to keep track of, if we answer a question differently, we will try to amend the ISO, but just understand that what is in the ISO is what governs. You can submit questions on the website, and we do post answers to the questions on the website. Register in SAM today. If you've never submitted to ARPA-H before, go to the submission portal and go ahead and get registered there, just in case. Because I will tell you, on submission day, everyone's going to be there. Which brings me to another piece of advice. Don't wait until it's time to submit. It is your responsibility to make sure your submission is received by ARPA-H by the due date and time. Paul told you earlier, if it's 10 minutes late, it's too late. I will tell you, if it's 1 minute late, it is too late. What happens a lot of times is within the last hour before submissions are due, everybody jumps on the site, and the site is only going to be able to handle so much at a time. So do not wait until the last minute to submit. And read the ISO. I think we have microphones for questions. So if you have questions specifically on my presentation, I will take them now, but you must speak into a microphone so our friends online can hear you.
Xianing, a professor from the Ohio State University College of Medicine. So my question is about review process. Can you talk a little bit more about how that is done? Is going to be by a review panel, or it's done by staff or program managers from ARPA-H? Thank you.
Yeah, the proposals are evaluated. There is an evaluation team that's put together. So it consists of program managers from ARPA-H. It could consist of other government employees from other government agencies who have the right expertise to evaluate. We also do have subject matter experts at ARPA-H who are contractors supporting the PMs, and while they don't evaluate your proposals, they will review them and provide their subject matter expertise to the PMs as the PMs consider which proposals to select.
Hi, I'm Bing Lin from New York Genome Center. My question is follow up the previous one. What's the time frame that we expect for the review decision to be made?
We try to evaluate very quickly. There are a lot of moving pieces, as you can imagine, pulling a team together potentially across multiple agencies and varying schedules. I will tell you that my hopes and dreams are that we get IGoR awarded this calendar year, to give you some idea, but to break it, each step down into a time, it's going to be difficult for me, but we do try to evaluate as quickly as we can and get notifications out as soon as possible. For the solution summaries, we try to turn things around in a few weeks. Yeah, so. Oh, I'm sorry. Let me rephrase that and restate it into the microphone for those online. Paul said that we try to turn them around in about a week.
All right. Langley from Ohio State University, College of Medicine as well. Question about the budgeting. So there's a solution summary stage where we're going to propose a rough budget, right? And there's afterwards a more, say, formal budget. What's the flexibility can we have? From early stage, later stage budgeting? The early stage, like I said, is just to give us an idea of where you think your solution is going to fall, but there's no set, percentage that you have to stay within. If it's wildly different, right? Mm-hmm. If your solution summary is a $5 million solution and your proposal is a $25 million solution, I do expect in your budget, in the write-up portion of your budget that you explain, but we need your budget to be accurate in order for you to execute. Because once we're going to negotiate that price before award, mm-hmm. and you need to be able to work within that budget, right? So make the changes that you need to make sure your budget is realistic and appropriate. If we feel there's something that's not, that's where we get into negotiations with you, but there's no set range. And also team member, early stage summary solution, we may have a rough team, then later on we may want to expand a team. Is that also flexible as well, right? Okay. Yes. Thank you.
Hi, Gary Ahn from University of Vermont. Is there a period, if you get selected to move forward, where the task description document is revised and the budget is revised and all that prior to the actual negotiation step?
Sometimes. So sometimes we will notify you of selection and ask for revisions prior to negotiations. Sometimes what we want to do is we want to jump into negotiations and have everyone at the table and we go through everything in real time together. So it'll really depend on what our negotiation strategy ends up looking like for IGoR. And at this time, I can't speak specifically to how we're going to handle negotiations.
Hi, I'm Shaheen Mohammadi from Cytognosis. I will read ISO in more details, but for the budgets, do we need to have more accurate one for all three phases, or phase one which has better visibility? Because it seems that there are more uncertainty, especially with respect to collaborative spec post phase one, and I would appreciate if you can provide some details.
Yeah, so we do expect your budgets to cover the entire program, understanding that there are some unknowns. So what you have to do is just explain to us what assumptions that you've put into your budget to account for those unknowns. One of the things about OTs that's great, again, is our flexibility and our ability to move wherever the science takes us, right? So if as you are working through this program, you and Yoram feel that, hey, maybe we need to go in a different direction, with the approach, then we have the ability to do that. But we do expect your proposal, the entirety of your proposal, to cover the entirety of the program. We will negotiate the entirety of the program up front, and the award will cover the entire program.
Hi, thank you so much. I'm Allie, and I'm the CEO of a startup. There's a hard cap on the number of teams that are allowed for each application, and then if it's okay, if there's potential overlap between the TAs and the focal points of the teams, because some of them tie so beautifully together. So curious to hear your thoughts on that.
Yeah, there's no cap on the number of team members that you can have on your team. Your team needs to look however you think it needs to look in order to be successful in the program. As far as overlap, that is also fine, right? What you have to do as part of your proposal is explain to us who on your team is doing what and why, so that the evaluation team can determine if you have the right expertise, in the right places. But we are not dictating how your team looks. We're not dictating who does what on your team, how big your team is. That is entirely up to you.
University, and having gone through this process before, I know the teaming process is highly dynamic and can metastasize on you pretty quickly and things like that. So I wondered, at this stage, can you give us a back-of-the-envelope calculation of what funding appropriation, if that's even appropriate, that you're thinking of putting toward this, and then we can just divide by 3 and begin to extrapolate that in reasonable terms. You know what I mean? It would just really help at the end of the day to have some guidance that would sort of give you boundaries, for lack of a better term. So thank you very much.
Understood. Short answer is no. We really want to see what you bring to the table. Sometimes we are allowed to stretch our budgets a bit if we find that the solutions require that. Sometimes we are able to, if your proposal is too high, but is still determined to be selectable, to get your budget to fit what we can reasonably pay for. But what we need for you all to do is really put together the best solutions, and we don't want you trying to fit your solutions into a number. We want you to propose the best solutions, tell us how much it's going to cost, and then work with us from there.
I have a question about SAM registration.
Sure.
Do all the subs need to be registered on SAM, or is it sufficient for the prime to be registered on SAM?
So what I care about is that the prime is active in SAM. Sub-registration requirements will be up to the prime to determine if you need your subs registered and, based on how you operate.
Hi, I'm Johnny from Tahoe Therapeutics. My question is around the solution summary. How much information on the teams and who's going to be doing what do you want in that 5-pager? Because it's not super long.
What do you guys? Can we get a microphone to a PM?
For the abstracts, high level, who is there? You don't have to go into detail about every last person in their bio sketch.
Aaron Adler from BBN. I think you kind of answered this, but just to ask more specifically, a lot of the TA3 and TA4 things are more collaborative efforts across whoever's awarded towards the end. Should we just be making our best budget guesses on those for the later phases and adjust as needed? Is that the approach you want us to take there?
Yeah, again, make your best guess. Let us know what assumptions you've made, because if we have insight into your thought process, then when we get to negotiations, we can also tell you where you might have misstepped and adjust at that point. And then again, as we get closer to those, the later phases of the program, you all in collaboration with the PM will determine where things are going to go. And, we'll have to have conversations about whether or not pricing needs to be adjusted and what we can or can't do at that point.
Over here. Hi, Marty Tristani, University of Utah. I promise I'll reread the ISO, but my first read of it, with respect to the phases 1, 2, and 3, my understanding was phase 1, 2, and 3 were different domains of a disease. And then this morning, hearing the sort of standards guy speak, it sounded like phases 1, 2, and 3 were different experimental expansion processes.
So what I'm going to encourage you to do is take one of those cards that we have, write that question, give it to Renee at the front, and we will tackle that in the afternoon Q&A.
Okay, great.
Hi, Leonardo Garcia, University of Pennsylvania. Sort of building off the other question about the collaborator. If we are working with, say, a research consortium or a disease consortium, would the award go to a single institute that would be responsible for dispersing to the relevant collaborators, or would it go to a multi-institutional allotment?
The former.
Hi, thank you for your talk. Natalie Ma, CBO at Deep Origin. This might get pushed to the afternoon, which is totally fine, but it sounds like that you want one marketplace in the end from the morning slides. That was a little unclear initially, at least from the ISO, because there were 2 places in which one suggested one marketplace, one suggested each team would have its own marketplace. So can we just get a understanding of what the intention is there?
Afternoon. Yeah, if you could submit that and we'll tackle it this afternoon.
Phil Barrett, Acquire, New York. Just following up on the question from over there. Can a proposal involve more than one TA1 element, more than one TA2 element? If those elements use different approaches but are complementary, or is there just supposed to be essentially one underlying model, one underlying approach to each proposal?
So each solution really needs to have one focused approach. If you have multiple team members who need to work together to accomplish TA1 or to accomplish TA2 or what have you, that's perfectly fine. But Paul and Yoram can correct me if I'm wrong here, but we're not looking for multiple different solutions in one proposal.
We can address this more fully. I mean, we're open to risk mitigation, right? And we think we're going to A, but we're also going to try B, right? But at some point you have to pick A or B, right? We don't want 10 different solutions. Ultimately, at the end of the day, you got to come up with a solution, but you can try different things as you go forward. Let's address it in the afternoon, and we'll revisit that. Yep.
Hi, Adrian Jabolski from Anubio. Is there a cap on indirect cost?
No. We don't, you need to propose either, if you have negotiated rates, right, propose those, otherwise propose what makes sense and then justify them to us.
Hello. Yeah, sorry. Yao Yang here from Cypher AI. My question is, can we like add, can like Prime add subcontractors later after proposal, or is it fixed?
Yeah, you can change your team as the work requires. However, you have to submit a proposal that demonstrates you can complete the entire program with the team that you've submitted. But we have had instances where, someone's picked up a new sub or swapped out subs. If that's required, you can do that. Understand though that in those circumstances, typically the budget is fixed, right? So we're not necessarily going to provide extra money. I mean, there are some exceptions to that, but for the most part, we expect you to be able to present a complete solution in your proposal and execute on that. But you don't have to keep your team static the entire program if that's not working for you.
Gotcha. Thank you.
Sorry, Gary on again. I think I know the answer to this, but I just wanted to confirm that the HHS salary caps are enforced for this?
Correct.
Okay.
And that is, for those who are unfamiliar, that is the OPM executive level 2 salary for this year. Salaries that ARPA-H is paying for under these awards cannot exceed that number.
I have Svi Goldenberg, Talzera. My question is, if you don't have a team, will you act as a matchmaker in case you like the proposal?
No. That's why we're here today, right? We have the poster sessions so that we have the lightning talks this afternoon. We have our teaming page. This is kind of how we are facilitating, but we don't match post-submission.
Dwight Baker, Selenome. As far as TA1s, if we have 2 different disease models that we want to partner with on that side, like if we had oncology and neurology, are we able to bring 2 different models forward?
We'll take that in the afternoon Q&A.
Hi, Laura Winterkorn, New York Genome Center. I don't see a budget justification included in the solution summary template. Do you want that information?
Nope.
No, not at all. Great, thanks.
Hi, a follow-up question regarding the prime responsibilities, because you mentioned that's going to be the one signing, but what are the expectations, if any, that who would be more appropriate, and what are the expectations from, like, let's say, the prime versus subs? Are there any expectations or any context that was not necessarily addressed? Thank you.
Sure. The biggest expectation that we have of the primes is that you can effectively manage the team, right? So the government is going to directly interact with that entity throughout the program, right? That's who we have privity of contract with. So we need you all to be able to manage your subs and manage all of the parts that are coming together in this program. You will be responsible for the disbursement of payment to your subs and whatever arrangement you have worked out. You are responsible for ensuring that your subs are executing on schedule, right, and meeting the required metrics. Now, one caveat to me saying that we're going to interact with the prime, that doesn't mean we won't have any interaction with your subs, right? I would bet that most of the meetings you are going to have with Yoram are going to include your entire team so that everybody can speak to their area of expertise. But we do expect the prime to be that intermediary. Prior to award, we negotiate with the prime, right? So if there are issues that we have with a subs proposal or changes that we need, then we filter all of that through the prime. So that will be your responsibility prior to award.
Aaron Adler from BBN. A question there was in the ISO clarification. Does all the software we develop need to be made publicly available? Like if it's funded by IGoR? There was a note in the ISO about making software publicly available. I didn't know if that was all software or not.
Don't remember. Yeah, would you allow us to answer that this afternoon so we can double check?
Hi, Siping Han, CEO, Apply AI.
My question is, are there any issues with subcontractors being part of more than one proposal? And is there any issue with subcontractors who end up selected in more than one team?
The short answer to that is no issues. However, we won't fund the same work twice. So if someone is a sub on multiple teams and they're doing the same work, right, and providing the same, the same outputs to each team, then what we're going to do is essentially we'll allow you to work on multiple teams, but we're not going to pay for that multiple times. If someone's a sub on multiple teams and you're doing different work, right, and different outputs, that's a different story. What we will still do in that case, though, is look to see if there are areas of overlap, and then we'll negotiate that as appropriate.
Hello, Sean Mannion, Praxis Science in Duquesne University. Similar to that question, can one group be a prime and also a sub on a separate one, both for proposal and also for execution? Thank you.
I think we said yes in the ISO to that. I believe the answer is yes, but I will double check and make sure that we get you a solid answer this afternoon.
Emek Demir, OHSU. There's heavy emphasis on the scalability, which makes kind of predicting the budget very, very difficult. Is it okay to provide a budget description that is on a per unit basis for some components of or do we always have to give a final number?
So we need to see your total price, right? We'll need to see in your proposal the total price that you're asking the government to pay, so that we can include, we have to, when we determine who's selectable and who fits the program, we have to be able to justify the total budget that we're planning on dispersing.
The challenges, for example, if we were to collaborate with, right, we have no ability to guess what the scale. Right, so that is kind of one of the big question marks in my mind.
Understood. So what I would ask you to do is make some assumptions, price based on those assumptions, and then when we get closer to that stage of actual execution, we'll reevaluate with you and determine if there's some mismatch in your budget, and we'll work from there. Yeah, Paul says for those online, we can address this in more detail this afternoon.
Susan Buckett-White, Ginkgo Bioworks. Specifically on the multiple
are selected, then we'll work with you to cut your budget down from there. But we need to see what each full budget is going to look like up front.
One tiny clarification. Sure. Would unique experiments for different teams be considered non-duplicative? One set of experiments over here, is that considered non-duplicative? Yes. Okay.
Yeah. Mm-hmm.
Just to make a clarification about the marketplace, I mean, the understanding is that in phase 3, if somebody, if team A orders from a lab that used to be on team C, they would pay for that. Right. And so it's not like you have to, if you're the one doing the research, you have to do it for free because they're in the program, like they would pay for it. And that's the beginning of how we're going to set pricing, right? Because I'm doing work for somebody else. And so we're going to, where you're saying there needs to be some unit pricing. And so that's the whole process. It's not like you're giving it for doing that work for free from this random other team. But I think a few questions are trending along that way.
Hi, Genevieve Steiner-Ryan from Johns Hopkins. So I pulled up the solutions submission page, and I'm literally on here, and there's a question about human subjects research. And given that, we're not allowed to collect new samples, do you consider existing patient lines human subjects research?
Yeah, sorry, actually, this came up in the FAQ. You're allowed to use existing samples. You cannot prospectively grab new samples.
And would you consider existing samples human subjects research?
That still falls on HSR.
Okay, perfect. Thank you.
Any other questions? One more.
And a quick follow-up on the samples question. Is it okay, for example, to rely on commercially available samples or biobank samples? Yes.
Yes.
Anyone else?
No more questions. I will be around for the remainder of the Proposers Day, so if you'd like to ask other questions, we do have the afternoon session, but if you have something that is really specific to your organization, feel free to grab me during a break, and happy to talk through it with you. Thank you.
Thank you so much, Adria. I think you answered questions for like twice as long as you spoke. That's fantastic. And Adria is really nice, but she has been on the ship way too many times as people are running down the dock trying to get to the ship and just waving to them. It's like, what can they do? So I believe she had some advice along the lines of read the ISO, right? Thanks to everyone for submitting your questions, and let's give you as many answers as we can. We condensed a lot of them. If you feel like we didn't fully cover a question, the answer, just hit us back up, and we will make sure that we answered in the FAQs. All of these are also going to appear in the FAQs. All the answers this morning from Adria, they're going to show up in the FAQs, so they're always good to review. All right, so teaming this. For TA4, is it acceptable to name a second lab node that is onboarded and validated during the phase 1 period of performance? Or must 2 validated nodes be in place at the time of solution summary submission? So 2 performing labs should be in place by the time a full proposal is submitted. We also encourage 2 at the abstract phase.
So the second question, many multimodal experimental packages have no single CRO that runs every assay. Labs specialize by modality. Does the 2 validated lab requirement expect both labs to perform the same assays, parentheses, so cross-lab reproducibility can be checked head-to-head, or can the 2 labs be complementary, each contributing a different modality that the requesting team assembles into the full package? And characteristically for ARPA-H, we took that gigantic question and said, the 2 labs must have at least one modality in common so that the interlab variability can be assessed. You'll see this a lot in the FAQs. We'll get these gigantic questions and the answers will be fairly short.
Question number 3, do the TAs need to have sole ownership? That is, some of the vendors we're approaching have capacity to do work in both TA1 and TA2. Is it preferable to have a vendor or a partner in our team only focus on a single TA, or is it okay to have cross-TA partners? Similar point for TA3 and TA4. The cloud labs have very useful input for both TA3 and TA4. No, I mean, the organization of the teams is up to the PI. You set them up however you want them. Over. Maybe just, yeah.
The solicitation mentions 4 examples of complex diseases, neurodegenerative conditions, chronic autoimmune disorders, complex pain syndromes, and emerging infectious diseases. Should we interpret that as a preferred focus on these areas? Would other areas such as understanding the tumor microenvironment be out of scope? Proposers, and the answer is proposers can propose any complex disease that they feel is amenable to the approach being developed by IGoR.
All right, question number 5. Would ARPA-H consider grouping multiple related diseases under a single disease to be consistent with the intent of the solicitation, with additional related diseases serving as the related second disease in phase 3? Or does ARPA-H expect each named disease to be considered a separate disease for program purposes? So we cannot comment on if a specific disease within scope, however, diseases that within the same disease ecosystem are certainly within scope.
Number 6, can banked human samples be used to generate new data based on the proposed experiment? For example, in TA3, the answer is yes, and this qualifies as human subjects research. Please refer to the ISO for limitations on AI and HSR and vertebrate research.
Question number 7, is it possible to form teams across academic institutions and pharma companies? Yes.
Number 8, how will the teams be evaluated? Or will all teams advance all phases once selected? Will teams define their own measures of success, or will the PM team define that? The answer is the ISO sets out milestones that are common across all teams. Proposers are encouraged to include metrics that are specific to their effort. Number 9.
Thanks you scrolled up for me. Thank you. Nice.
That way.
You're a nice guy. Does all software developed with ARPA-H funding need to be made publicly available? So a key objective of IGoR are open standards that allow interoperability between a variety of laboratories. At the same time, we fully realize that some aspects will be proprietary. We want you to hold on to those to ensure commercialization, right? So the real focus is on the standards and ensured interoperability. Over.
So number 10, one of the metrics counts, quote, exceptions per experiment. If we have a human move a plate from one machine to another, and this is planned, is that an exception versus fixing a step that was intended to be automated? And the answer is if a step is intended to be run by a human and it runs successfully, it is not an exception.
Question number 11, can ARPA-H please clarify whether 3 distinct lab types, cloud, automated, standard, are required or desired for TA4 or is more than one lab with the same type? Okay. Answer, proposers may include any mixture of lab types that they feel is most efficient for the problem.
Okay, number 12, does IGoR have the same expectation of reproducibility on computational models and AI platforms? The answer is no. The key focus is on reproducible experimentation. But I should add a caveat that we can talk about IV and V for models maybe at the end of this if there's a moment.
Yeah. Yes.
If there's a moment.
It's not in the ISO, but it's something that you're interested in. So that's,
yes.
Okay. Can you elaborate on how generalizable the approach should be to other disease areas when one is proposed? Should methods, reference materials generalize to other areas or be focused to one disease? So techniques should be generalizable to the greatest extent possible, right? That is something that we're looking at, right? Both to address multiple diseases, but also to establish a viable marketplace. If you are generating, some technique that is so refined to a rare disease, it's going to be very hard to generate a viable company based on that.
Number 14, do TA3 experiments need to currently exist? Is there room for new development? There is room for new development.
There you go. For TA4, there's a trade-off between breadth of experiment, capability coverage versus reproducibility. Is there an emphasis on one versus the other? Teams should propose TA4 repertoire that adequately cover key questions in the proposed disease area. It is not expected that all experiments will be automated.
Number 16, it sounds like teams in TA4 will need to perform TA3 protocols from other teams. Is this correct? Yes.
17, how distinct disease areas need to be? For example, should latter phases be in a different organ or entire focus area? Answer, it can be a related disease of the same organ or the same etiology.
Number 18, is there any preference for technical focus, for example, early versus late stage drug development? The answer is the expected output would be a mechanistic model of a disease that would enable the development of new therapies.
Question number 19, how to validate complex mechanisms if experiments are limited to simple in vitro models? Answer, proposers should propose diseases amenable to understanding from cellular to multicellular assays. Performers can also use invertebrate animals and more complex tissue cultures. Higher animal models are still allowed at the discretion of the human researcher.
I could add just a small thing to that, even if it isn't directly in the FAQ. The simplest way to get around that question is connect to a model that also predicts what will happen at a higher up scale. And that's how you can get into that exact answer that Paul just read. Number 20, how do you envision the marketplace to run in the long term in terms of cost reimbursement, intellectual property, and scientific presentation publication? The answer is we expect that the proto marketplace will evolve into a fully formed marketplace where companies are reimbursed for the services provided.
Question number 21, clarification on mechanistic. Can this be causal or does it require a biophysical component? The model should be causal and does not require biophysical components.
Number 22, how much of the proposed experimental procedures must be interoperable at the 2 proposed labs? And the answer is proposers should identify to what extent they will have overlap, which should be as great as possible.
Question number 23, is the marketplace intended to be primarily a financially facilitated platform, experiments for cash, or should other Mortonian plus non-financial elements of science incentive such as puzzle solving factor into this economy? Experiments for cash.
Which coincidentally enough was literally the sign on my door in my lab. Number 24, how well integrated do the actors in each TA need to be? For example, do the TA1 researchers need to be human as a loop for TA2/3 elements and participate in the market of TA4? Or is integration across the TAs a nice to have? And the answer is integration across TAs are central to the program. Central.
Question number 25. Can the computability requirement for TA1 be explained more? Do the models have to support mechanism-based numerical simulations, or would any model that give deterministic predictions work? So answer is we are open to a range of models that capture both complexity and causality.
Number 26. Does TA4 require that all experiments be fully automated? Can protocols include manual processes? And the answer is no, not using robotics. Yes, they can include manual processes.
Question number 27. For TA4, what does an automated marketplace mean? Does it need to be intelligent? Do CRO reference labs count? CROs and reference labs count?
Number 28, TA1 excludes the development of new foundation models such as training a new LLM from scratch. Can you provide a more precise definition of foundation model? For TA1, we will not support the development of models that are predominantly data-driven, where the gathering or training of data is the main expense.
All right, and the final question, when is your Proposers Day being held? Today. Yeah, I think you guys all got that. All right, that's it. And I think you had some final comments.
Well, so the question had come up, and there was a nice lightning talk about it, about IV&V, and that there's really more to IV&V than what it says in the ISO. We fully know that, and the intent is to come back to that at a later stage, but to look at IV&V for the models as well, so that stay tuned is the best I would say. But with regards to the what's in the ISO, as Adria said, read the ISO. For IGoR, we are just expecting what it says to happen as what's currently stated in the ISO. Okay, so thank you very much again, and I think this concludes the event, if I'm correct. So again, I just want to thank everyone that came and sticking it out till the end. I hope that this has been, if nothing else, a really great networking event for you. It was really nice to see a lot of interaction. That's part of what we hope for anyway, is that realistically, it's impossible to be able to fund every single person that's in this room and the others that are online and that will be submitting things. That's an obvious practical impossibility. But if we can help drive change just through the interaction and through the fact that this is moving things into a new direction, then I think we can be really, really happy with the outcome. So again, thank you all. And if you have further questions, please do submit them to the FAQs. This often happens after Proposers Day. And best of luck. Thank you.